In this video, we're going to talk about, begin our discussion of cell respiration, and this is sections 3.7 and 8.1 in the IB standard. And respiration is really the process of taking sugar and turning that into a usable form. Organisms do this, we do this. Organisms need energy, and that energy, humans, for example, get that by eating different foods, plants and animals, and our bodies and other organisms take that energy and convert it into a form of energy that the cells can use called ATP. And so respiration, cellular respiration, we're looking at how this occurs, and in part one of the series here we're going to look at the first kind of steps of this. So in an introduction to cellular respiration is, the definition of it is that it's a controlled release of energy from organic compounds and cells to form ATP. And the important part here that we're looking at is this controlled release. It doesn't happen all at once. It's kind of a slow breakdown of this energy to form ATP. So it's not a boom all of a sudden, it's a slow controlled release. In cellular respiration, glucose in the cytoplasm is broken down by the process of glycolysis that we'll look at today into a molecule called pyruvate. And this process has a very small yield of ATP. The overall process we'll see of cellular respiration produces lots of ATP. This first portion, called glycolysis, produces only a small yield of ATP. And this process occurs in the mitochondria. And so the first process of glycolysis actually occurs in the cytoplasm, but the overall process of cellular respiration and the production of most of the ATP is occurring in the mitochondria. And let's take a closer look at the mitochondria, and I'll put an image of this on my website, so if you can't see it very well here, you'll be able to. This is an electron micrograph of a mitochondria, and it's kind of hard to make out here, but there's an outer and an inner membrane. And so the outer membrane is the outermost portion of the membrane of the mitochondria, just like the cell membrane has a membrane, an outer portion, inner portion, so does the mitochondria. And so there's an outer and inner portion. These little projections here, these are kind of like little folds or flaps, kind of like in our stomachs we have. It's not a nice flat surface. It goes up and down, up and down, up and down. And these in the mitochondria are called cristae. And the purpose of these is to increase the surface area. The process of cellular respiration is going to occur in the mitochondria, and it uses the different membranes in order for the process to actually take place. And if there's more surface area, more of these reactions can occur. And so these cristae actually provide that increase of surface area. The space, the open space, or kind of the cytoplasm of the mitochondria, we call the matrix. And then the intermembrane space is in between the different cristae and the different portions of the mitochondria. Before we actually kind of get into the process, as more of an introduction, let's look at the inputs and the outputs of cellular respiration as a whole. This process uses one glucose, so we're using one glucose molecule, and the molecular formula for that is C6H12O6, and then we use 6 oxygen atoms, O2. As a result of this process, or what's produced, we get 6 CO2, 6 H2O, 6 water molecules, and energy in the form of ATP. Now, through and during this process, we're going to see different molecules gain and lose electrons. And there's a specific description, a specific saying of how this occurs, and that's oxidation and reduction. And so during cellular respiration, the process results in the oxidation of some elements and the reduction of others. Now, maybe you remember this from chemistry, hopefully. Oxidation refers to the loss of electrons from a substance. And this can occur by either gaining oxygen or losing hydrogen. The opposite of that is reduction. If a molecule is reduced, it has the addition of electrons to a substance. And so this could be losing oxygen or gaining hydrogen. Either of those could be occurring. And so here's an image to kind of better represent that. Reduced compound A is the reducing agent. The compound A is oxidized and it loses electrons, and so that is oxidized. The oxidized compound B, on this side over here, B is reduced and it gains electrons, and so compound B is reduced. So now let's actually take a look at and see this process of glycolysis. This is a pretty complex process. There's a number of steps to it, and I'm going to walk through those verbally and then also give you some steps listed out that you'll be able to have as well. And I'll put this image on my website as well so that you can go back and look at it if you'd like. It's just from Wikipedia. Glycolysis starts with a single sugar molecule. Most oftentimes this is glucose. It could be basically any 6-carbon sugar, a hexose sugar, but we're going to look at and talk about it with glucose in this example. And really what's happening here in our first steps is a phosphate from ATP is transferred to our glucose molecule. So here's our glucose. We've gotten an additional ATP molecule, and it makes our glucose molecule more reactive. And it also converts it to fructose in this case. So we have our fructose here. The enzyme phosphofructokinase is going to transfer a second phosphate to the fructose molecule from ATP. So we're using 2 ATPs here. Here's our first one, here's our second one. Now this is an investment of energy. It would seem like it's maybe counterproductive to actually use ATP to build ATP, but eventually through the whole process we're going to have a larger net yield of ATP, as we'll see as we get farther through cellular respiration. So right now we have this very reactive, this fructose molecule that has had 2 additional phosphates on it. And a second enzyme called aldolase is going to split or cleave this 6-carbon sugar into 2 3-carbon sugars. We have 2 separate 3-carbon sugars. And those 2 3-carbon sugars are going to be oxidized. They lose some electrons, and that occurs by removing a hydrogen molecule from each. So we've lost some hydrogen molecules from each of these 3-carbon sugars at this point right here. Now, we're going to eventually we're going to walk through this. We'll walk through this process, and we're just going to explain it for one molecule, obviously, but it happens for both at the same time. And so now that we have our 2 of our 3-carbon sugars, these excess hydrogen ions that have been released are going to be combined with something called NADH. So we're going to make NAD. They're going to be combined with NAD plus, which is an energy molecule that will be used a little bit later on in the process, and this NAD is going to accept those hydrogen ions, so it's going to produce NADH. And then in our following steps here, each of these 3-carbon sugars are going to lose a phosphorus, and so that's combined with ADP. So that ADP is adenine diphosphate. An additional phosphate from the 3-carbon sugar is added to that to make ATP. And this is going to happen twice for each molecule, 1, 2. So we're going to produce 2 ATP per 3-carbon molecule that we start with. So overall, this is going to produce 4 ATP molecules. Now we've spent 2 ATP molecules to get from our glucose to the fructose. So overall, our net gain from glycolysis is 2 ATP sugars. Let's look through the steps here. As we discussed, one phosphate from ATP is transferred to glucose, or another 6-carbon sugar. That glucose is converted to fructose. The enzyme transfers a second phosphate to fructose molecule from ATP. The enzyme cleaves the sugar into 2 different 3-carbon sugars. Each 3-carbon sugar molecule is oxidized, meaning it loses electrons by removing 2 hydrogen molecules from each of our 3-carbon sugars, and 1 phosphate is attached to each 3-carbon sugar. NAD plus accepts 1 hydrogen from each of the 3-carbon sugars to create NADH, and 2 hydrogen ions are produced. Both of those will be used a little bit later in our electron transport chain. And then 2 phosphates from step 5 from each molecule are added to ADP to make ATP. So overall, through this process, it occurs in the cytoplasm. Our inputs, we input glucose or some other 6-carbon hexose sugar. 2 ATP molecules are produced, our input, and 2 NAD plus. It's a positively charged molecule, our input. Our outputs, on the other hand, are 2 pyruvates. Those are the 3-carbon sugars, and we get 2 waters from this process. We get a net of 2 ATP, so we've got a positive 2 ATP. And we get 2 NADH and 2 hydrogen ions. This ATP is going to be obviously useful for the cell. The pyruvate is what's going to be used in our next portion to actually continue cellular respiration, and the NADH and 2 hydrogen ions will be used during the electron transport chain portion of cellular respiration. In our next video, we'll look more specifically at this process and the details of the electron transport chain and how we get a high yield of ATP.